Now that we've gone through graded potentials and action potentials, we're going to take some time to discuss how a signal gets from one neuron to another. In this video, we're going to talk about synapses. We're going to talk about chemical synapse and electrical synapse. We're going to talk about neurotransmitters, add some more information to EPSPs and IPSPs, and then we're going to wrap it all up with synaptic potentiation. The easiest way to define a synapse is just a region where a neuron is going to pass a signal on to another cell. Often that other cell is a neuron, but in the peripheral nervous system, it may be a muscle or a gland as well. We're going to discuss electrical synapses and chemical synapses. We're going to start with electrical synapses just because they are the simplest. With an electrical synapse, the cells that are communicating are very close together. So we have, here's the presynaptic neuron and here's the postsynaptic neuron, and they're held together by these gap junctions. And gap junctions allow for electrical signals to go from one cell to the next. And so these are very, very quick signals, and the information can actually go in both directions, so it's bidirectional. We see a lot of electrical synapses in embryonic tissue. In adults, we see electrical synapses in neurons that are involved in eye movement and in motions and memory. We're going to focus mostly on the chemical synapse. The chemical synapse is the most common. It only allows unidirectional information transmission. So information only comes down from the presynaptic cell to the postsynaptic cell. The chemical that's involved are called neurotransmitters, and neurotransmitters is a class of chemicals that includes a very broad array of chemicals, including things like carbon dioxide and epinephrine and opioids, all these very diverse chemicals. So when that action potential travels down the axon and finally gets to the axon terminal, right down here on this axon terminal, there's one more voltage-gated channel that I haven't talked about yet. And these are voltage-gated calcium channels. When that action potential gets here, it triggers the opening of these calcium channels, and calcium goes rushing inside the axon terminal. The calcium rushing inside tells these synaptic vesicles that they're going to release their neurotransmitter, or that they should release their neurotransmitter. And so the vesicles are going to release that neurotransmitter by exocytosis. So the membrane that holds the neurotransmitter together in that vesicle is going to merge with the plasma membrane of the cell, and it's going to kind of inside itself out and then release neurotransmitters into this region called the synaptic cleft. Okay, this takes some time, and so the time that it takes for the neurotransmitter to be released and travel across the cleft is called a synaptic delay. So with an electrical synapse, there is no delay. That electrical signal goes straight through to another cell. With a chemical synapse, there's a little bit of a delay, but it's not a very long delay when you're thinking about it. And so once that neurotransmitter floats around the synaptic cleft and eventually binds to a receptor on the postsynaptic cell, it's going to create a graded potential on the postsynaptic cell. And we talked about those in a previous video, so you can review the graded potential video if you would like. So just a little bit more detail about neurotransmitters. As I mentioned before, they're chemical messengers. Neurotransmitters can target neurons, muscles, or glands. It all depends on whether or not that cell has a receptor for the neurotransmitter. In addition, neurotransmitters can have different effects on different cells. So sometimes a neurotransmitter can excite a cell, and sometimes it can inhibit a cell. And so that can happen with the same neurotransmitter. The effect of a neurotransmitter has to be stopped eventually, though, right? You don't want to keep that neurotransmitter in between the synaptic cleft, right? Because then neurons would be getting excited or inhibited even though there wasn't like a new stimulus there. So the way that that happens, the way that neurotransmitters are stopped, is either by reuptake by either a neuron or a neuroglial cell, or sometimes the neurotransmitter is broken down by an enzyme, like acetylcholine is broken down by an enzyme called acetylcholine esterase. And the other way is just simple diffusion, right? There's nothing keeping the neurotransmitters in that synaptic cleft. And so over time, the neurotransmitters are just going to diffuse away. There are many different types of neurotransmitters, and our understanding of what chemicals can act as neurotransmitters is growing each year. Some of the most common neurotransmitters are shown here. We're going to start with, we have adrenaline and noradrenaline. You've also heard these called epinephrine and norepinephrine, and they are involved in the fight or flight response. We have dopamine, and dopamine is a neurotransmitter that helps us feel pleasure. And so that can be pleasure from food, from experience, from winning the lottery. All of these things give you little dopamine bursts. Then we have serotonin. Serotonin's involved in digestion and sleep, and serotonin is directly affected by the amount of exercise that we have and the amount of light that we're exposed to. There's GABA, acetylcholine, glutamate, and then kind of the class of endorphins. And these have pretty broad-ranging effects depending on what the target cell is. We're going to focus mostly on acetylcholine, epinephrine, and norepinephrine throughout the rest of our studies in the nervous system. I mentioned that neurotransmitters can have an excitatory response or inhibitory response in the postsynaptic cell. And so this is all kind of dependent on the specific synapse. And so if we're looking at an excitatory synapse, when the receptor gets its neurotransmitter, the neurotransmitter binds to the receptor. In an excitatory synapse, there's going to be a sodium influx and a potassium efflux. And so this depolarizes the membrane. Remember, these are the same ionic movements that happen along the axon during an action potential. So if this is going to happen on the receptive end, on the dendrite and soma end, this is still going to depolarize the membrane. It's just going to be a local depolarization. When that happens, it's an EPSP, right? An excitatory postsynaptic potential. If enough EPSPs come together and they can overcome any of the inhibitory signals, and it reaches a threshold at negative 55 millivolts, you get an action potential. The alternative is an inhibitory synapse. With an inhibitory synapse, one of two things happen. Either potassium channels open and potassium moves out of the cell, or the chloride channels open and chloride moves into a cell. This is going to cause hyperpolarization, and that's going to reduce the postsynaptic neuron's ability to have an action potential. I think that after all the work that we've done with action potentials, if you sit down and kind of sketch out what happens here and maybe try to graph it like we graphed the action potential in the last video, that would help you really kind of solidify the excitatory response and inhibitory response that we see at these synapses. I hope this video is helped clarifying what happens at a synapse and the different effects of neurotransmitters. Again, if you have questions, please post to the Community Help Forum, and I will answer them as soon as possible. Thanks so much. Take care.